Today we'll be discussing translation process in eukaryotes. Translation involves translating the sequence of a messenger RNA molecule to a sequence of amino acids during protein synthesis. Ribosomes in the cytoplasm or endoplasmic reticulum synthesize proteins after the process of transcription of DNA to RNA. Translation takes place in three stages: enunciation, elongation, and termination. Now, before starting with the translation process, I just want to tell you about the tools of translation. So tools of translation includes ribosomes, amino acids, tRNA, mRNA, factors, and enzymes. Ribosomes exist normally as separate subunits that are composed of proteins and rRNA. Eukaryotic ribosomes are larger and ATS type, and they are more complex as compared to the prokaryotic ribosomes. I can see here this is the structure of ATS ribosome, and ATS ribosomes having two subunits, larger 60S subunit and smaller 40S subunit. 60S subunit is made of 28S rRNA, 5.8S rRNA, 5S rRNA plus 49 proteins. 40S subunit is made of 18S rRNA plus 33 different proteins. These subunits come together to form a ribosome. When they bind to an mRNA near its 5' end. On binding to an mRNA, the ribosome reads the nucleotide sequence from the 5' to 3' direction, synthesizing the corresponding protein from amino acids in an N-terminal to C-terminal direction. Each ribosome has three binding sites for tRNAs: A site, P site, and E site. A site means amino acyl tRNA binding site. It's a site where during elongation the incoming amino acyl tRNA binds. Then P site, peptidyl tRNA binding site, is the site where the tRNA linked to the growing polypeptide chain is bound. Then exit site, that is E site, it's a site for tRNA following its role in translation and prior to its release from the ribosome. Then mRNA. mRNA acts as a carrier molecule. So it carries the genetic information transcribed from DNA. It contains a sequence of 3 nucleotide units called codons, which specify the order of amino acids in the protein chain. Then tRNA. tRNA is an adapter molecule that brings specific amino acids to the ribosome, matching mRNA codon with anticodons. And amino acids, we know the amino acids are the building blocks of the protein. Then factors and enzymes. There are three types of different factors that are involved in translation process: are enunciation factors, elongation factors, and termination factors. And two major enzymes which are involved in this process are amino acyl tRNA synthetases and peptidyl transferase. Now, translation in eukaryotes. So first step of translation is enunciation of protein synthesis. So in eukaryotes, the small subunit is already associated with the initiated tRNA when it is recruited to the capped 5' end of the mRNA. It then scans along the mRNA in 5 to 3 direction until it reaches the first start codon, means first EUG, which is it recognizes as the start codon. So eukaryotic cells require more auxiliary proteins to drive the enunciation process as compared to the prokaryotes. So this enunciation process can be divided into different steps. Like number one, binding of the initiated tRNA to the small subunit always proceeds association with the mRNA. A separate set of auxiliary factors mediate the recognition of mRNA. Then small ribosomal subunit bound to the initiated tRNA scan the mRNA for the first AUG sequence. AUG is a start codon sequence. So large subunit of the ribosome is then recruited after the initiated tRNA base pairs with the start codon. So, initiation factors. Just look at this diagram. This is smaller subunit of this is smaller subunit of the ribosome, larger subunit and smaller subunit. On the larger subunit, these three sites, A site, P site and E site. So on the this smaller subunit, initiation factors EIF1, just look at this right there, EIF1, EIF1A, EIF3 and EIF5 bind to the small subunit of the ribosome. EIF1, EIF1A and EIF5 acted together to prevent both large subunit binding and tRNA binding to the A site. Then initiator tRNA is escorted to this small subunit by 3 subunit GTP binding protein EIF2. You can see here this part. So EIF2 binds the initiator tRNA only in the GTP bound state, right? The complex between the initiator tRNA and EIF2 is called ternary complex TC. The initiated tRNA is charged with methionine because in the case of eukaryotes, tRNA is charged with methionine, not with N-formyl methionine as in the case of prokaryotes. So it's referred to as methionine tRNA methionine complex. So EIF2 then positions this methionine tRNA methionine in the P site of the initiation factor bound small subunit, resulting in the formation of 43S pre-initiation complex. So you can see here, this is 43S pre-initiation complex, 43S PIC. Next step is elongation. Once the peptidyl transferase reaction has occurred, the tRNA in the P site is deacylated. You can see here ribosome, A site, P site and E site. So tRNA in the P site is deacylated, means no longer attached to the amino acid, and the growing polypeptide chain is linked to the tRNA in the A site. This is A site. Now, for a new round of peptide chain formation to occur, the P site tRNA, it must move to the E site. And the A site tRNA must move to the P site. At the same time, the mRNA must move by 3 nucleotides to expose the next codon. So these movements are coordinated with the ribosome and are collectively referred to as translocation. The initial step of translocation are coupled to the peptidyl transferase reaction. So once the growing peptide chain has been transferred to the A site tRNA, and the A and P site tRNAs have a preference to occupy new positions in the larger subunit. So 3' end of A site tRNA is bound to the growing polypeptide chain and prefers to bind to the P site of the larger subunit. Is now decylated P site tRNA is no longer attached to the growing polypeptide chain and prefers to bind in the E site. You can see here, last diagram, E site of the larger subunit. In contrast, at this time, the anticodons of these tRNA remain in their initial location in the small subunit, bound to the mRNA. Now this A site is weakened and ready for the next incoming amino acid tRNA, and the cycle repeats. Next step and the last step is termination. Here, release factor, that is RF1 and RF2. First, ERF1 recognizes all the three stop codons and brings the GGQ motif into the peptidyl transferase center, leading to polypeptide release. So when stop codons are UAA, UAG, and UGA enters the A site of the ribosome, it is recognized by the release factor ERF1. ERF1 interacts with ERF3, which binds to a GTP-GTP complex, forming a quaternary complex. ERF1, ERF3, GTP, and magnesium ions. ERF3 then hydrolyzes GTP, which cause a conformational changes in the complex, and this change allows the middle domain of ERF1 to move into the peptidyl transferase center, where it catalyzes the hydrolysis of the bond between the polypeptide chain and the tRNA. So this releases the newly synthesized polypeptide chain from the ribosome. Now just look at the last part of this diagram. After the polypeptide chain is released, this is polypeptide chain. After this chain is released, the ribosome is disassembled, and the 60S ribosomal subunit is separated by the ATP binding cassette protein APCE1. Thank you.